Thanks very much, Paul. I think this has been a very, very useful summary. And first of all, a nomenclature on my part, I like to call it the WHO, because the WHO for me means a rock band. So, I mean, I like to really start out with commending the WHO, because I think the old system has outlived its days. And I think we all remember these pictures of an antibody with colored variable domains and then colored CDRs and then all in one color. Of course, life has become way more complicated, and there's, I mean, as Paul has very nicely explained, there are display technologies, there are transgenic animals, there's humanization with all methods, and there's all kinds of hybrid methods. So I think a lot of things are using multiple methods, and so clearly life has become much, much more complicated, and the whole thing is blurred. So there is a continuum between real human to humanized to chimeric, and this is sort of really a total continuum. I think that's the only way to describe it. So the method how an antibody is derived, that it was sort of in the old days a whole variable domain that's grafted to the rest of the chain, or just the CDRs, this is just obviously no longer true. So therefore, what the WHO said, which is that the sequence alone is the key thing, is good. Now, we have to, however, think this through, and the key question is, what's the purpose of all of this in the first place? And it was clearly to confer to the community some measure of humanness, probably implicitly saying some measure of immunogenicity. That is clearly behind these names. And so I think, as Paul said, the flaw with this is that if you make a sequence comparison, there is absolutely no justification why 85% is a magic number. Why is an antibody that is 86% identical way better than something that is 84%? So, and we can now, of course, discuss should this bar somewhere different, but I would suggest this bar doesn't make any sense at all. There is a gray zone, and there is no objective measure. Mankind doesn't know yet how immunogenic a particular molecule with a particular sequence will be. And so therefore, this definition would suggest a certainty that doesn't exist. And so therefore, I think there is a fundamental problem for drawing this line. And so you might say, well, let's call it all synthetic. That's one of the proposals, and differentiate it from human. But I think there's also a problem with that, as Paul has already said, which is that the human and the synthetic versions of this are also blurred. And so therefore, even that line cannot be drawn from the sequence alone. And so you might then say, and so clearly, I mean, I make that very, very clear. My proposal is to drop the substem completely. I think it has lived out its day. Then you might say, well, doesn't that open the door to things that are really very far away, and should they not be designated? I would say that's what the FDA is for. Things that are murine will never be approved again. So this is sort of an irrelevant problem. And so instead of haggling about what exactly to call into CDRs and where to set the threshold, I would clearly vote for getting rid of this, because no matter what you do, you will get intrinsic inconsistencies. So it's just an insolvable problem because it's a gray zone, it's a continuum, it just can't be done. And so, I mean, I would clearly propose that one should say to the WHO, it's well intended, time has moved, things have become much more complicated than CDRs or whole domains, and everything in between, and so therefore, you know, maybe we'll just get rid of it and call it MAP. And that is actually then also not inconsistent with the past.
Thank you, Andres. Would you like to comment, Marcus?
Yeah, I think it was important to us to get you all on board on this topic, because, as Andreas said, we or at least we at MorphoSys never cared a lot about this WHO naming. We had all these pictures in mind, and it was never an issue. The antibodies from the library always were fully human. And I disagree with Paul that this is a dry topic, because when we get 2 phone calls within a week from customers telling us, oh, there are the proposed names we handed into WHO were denied and the antibodies are now humanized. And so we started to dig in all the history, which you can see on the WHO webpage. So all the minutes from these meetings are public. And it was really nice to see. It's like going back in time and seeing how the technology evolved and how the WHO tried to keep pace with all this engineering efforts and all with this changing environment. So you find their protocols from Immunogen, who used humanization methods described in the same paper, and you will find that even the same method was used, but because other methods came up in the years, the first name was designated as a humanized version. The other methods, other humanization methods probably were developed in the time 2 years later. An antibody described in the very same paper then was designated as chimeric. Even the same method was applied. So it was really the WHO who tries to keep track with the evolving technologies and with the evolving engineering of the antibodies. And I completely agree with Andreas. It's difficult. How should this end? There will be new technologies more and more. None of the human antibodies, even isolated from B cells, will become a drug as it is. It will also always be engineered. You will remove the glycosylation site, or you will change the deamination site or whatever, and even simple changes, then is it still human or is it now synthetic or whatever? So it's really hard to determine this. And would we really like to have a bad antibody with a bad developability just to keep the fully human nomenclature? Probably we wouldn't, and nobody would do this. So it's completely ridiculous to stick to it. And also, what is the right way of alignment? So we started, of course, out to look what would be the right, or what would be the new nomenclature for antibodies for the lead candidates. We also handed out to our customers, and our experts internally came back, and everybody presented another homology, because however you do the alignment, you end up with 84. Oh, it's chimeric, or with 92. Oh, it's human. So it's hard to make the rules, and what are the rules good for? So they won't tell us anything here. So this is why I think we can completely agree with Andreas. It makes no sense anymore. Try to keep with the fast pace, and we are in the lucky position to have new ideas and to make better antibodies and to have new engineering techniques. It's just meaningless to try to always change the rules every half a year just to keep up with this.
Thank you. Max?
Yes, so, you stole a couple of my points, but they were all excellent points. I think the developability, the chemistry, I think as we learn more about it, is a very important point that we cannot compromise the quality based on real science, based on chemistry and biophysical properties, just to say, oh, now I'm going to drop to 84%. Oh my gosh, it doesn't work. We commented briefly during the presentation about the definition currently not taking the J region. Well, it doesn't take the whole constant region. And of course, in the field of bispecifics in particular, there is a lot of stuff that is going on in the constant region that's not taken into account. And so that just emphasizes the point that there are so many things, so many variations. I'm not advocating we have to have a rule for that. Obviously, just is a mess. And so Andreas' proposal initially of saying just dispense with everything, call it MABs, makes a lot of sense. Speaking of bispecifics, a number of people, for obvious reasons, are creating novel therapeutics based on existing modules. So as you can see, almost all the old antibodies are candidates to be a bispecific component, are now chimeric. So now what? So now I take Avastin and Herceptin, and now they're both humanized historically, but because they've been evaluated in 2016, now they become chimeric? It doesn't make sense. That's my statement.
Thanks, Max. Matthew?
I think the points have already been very well made about the flaws in the current system, so I'm not going to elaborate on what my colleagues have gone through already. There are just a couple of points I would make, though. The first is, as a scientist, I've never encountered a situation where everybody I've spoken to agrees. And this is the first time where every single organization and every single individual I've spoken to has actually agreed that this makes no sense whatsoever. The other point I would make is that, particularly from the humanization perspective, this artificial threshold that's been introduced really precludes us from using mouse germline sequences that are far away from human, because they will simply never obtain the zumab status, even through a straight CDR graft. So at present, we're having to pre-screen mouse germline sequences and exclude them even if they have the right function, simply because they would never achieve a zumab status. And that's a big concern, because these definitions are essentially hampering the development of therapeutics.
Great, thanks very much. And so what we're going to do now is to open the floor to any questions that you might have, or comments that you have in sort of encountering the kind of new system in real life. So we've already got several speakers. Please, in the middle there. We need some help with the mic in the middle. Good, thanks.
No, I think we're on now. So look, I would just like to agree that the 85% cutoff is ridiculous. But if there is identity as part of a definition, then as much attention needs to be paid to the germline repertoire. You mentioned the fact that the IMGT repertoire is evolving. In fact, at the moment, there are something like 100 polymorphisms that some of us recognise that IMGT doesn't. So I think it's important for people to realise the size of the argument within the community already over germline genes. And if you start talking about antibodies coming from other species, then that's a further nightmare, because the current definitions of germline genes in other species, I don't mean the ones that people are particularly using at the moment, but as we move into more exotic species, those species are having their germline genes defined, but I think being defined very poorly by reference to genomic reference sequences which have been poorly assembled. So the nightmare will only increase if that definition stands.
I agree. And I think, actually, if many of you think about sequencing antibodies from more species, maybe including more non-human primates, and kind of the depth of coverage to actually understand polymorphisms in other species, you realize that just becomes, what is already a headache, it just becomes a complete nightmare. Jim.
Okay, so the first thing is, I think most of the people in the room have a background in chemistry and would love to be able to name antibodies the way we name chemicals. Correct? Do you have a way to do that?
Well, I mean, if I have to enumerate all the amino acid sequence, it's going to be a very
Okay.
long name.
So this is a little bit of a Socratic dialogue. That's what we would like. We agree the old one is bad, but we'd like something like the IUPAC, whatever it is. The chemists know it better than I do. Yeah, UPAC. Okay. But that's probably not possible. So the answer comes in our cell phones. Okay. And what I would propose is that you name these by the year when they're registered. Okay. When I want to name, when I have an antibody that I think is worth naming, I get 2015 and they start numbering 1 through X. Okay. So you'll have the class of 2015, 2016, 2000, whatever. And if you want to know what it is, you pick up your cell phone, type in the number, and you'll get the sequence, and it'll tell you immediately whether it's human, mouse, or jackrabbit, whatever it is. Okay? Okay? It's real simple. You start, and actually you could use that and go back historically and rename all of the antibodies you have up there. Okay? And because the documentation of when they were first proposed with Herceptin. I'm sure Genentech talked to the WHO back in the middle 2000s, or in the middle 1990s, and there's a date there. So that antibody would be 1992-1 or whatever, okay? And some of these other antibodies would be 2015, 16, whatever, okay? And with the availability of our cell phones and the fact that we're all connected, within a few minutes or seconds, you would immediately be able to tell, type it in and know what it was. That's my proposal. Really simple. Gets rid of all the problem.
Thank you, Jim. I don't know if we had any comments, so we just want to move on to the next. I think in the interest of time, why don't we sort of move on to the next question in the middle so that we can have as many people sort of weigh in with comments, suggestions as the time allows.
Yeah, I'm from Novartis, and I had heard about the nomenclature change and didn't worry too much. And indeed, yesterday it hit us. So we got a comment from WHO back, and so we have one single chain which got the zumab substem, and now we have another one, and the frameworks differ in 3 amino acids, and now this they want to declare as chimeric. And the reason is just the frameworks are nearly identical. It's just the CDRs are completely different because we graft them from rabbits. So I fully agree that this system doesn't make sense anymore.
Great, thank you. Question at the front.
Yeah, this might be a bit leading for Matt, but anyone had any feedback from the INN committee as to why they've made the decision, how they defend it?
It's a great question. I don't know if anybody on the panel has any insights on that one.
I mean, we've attended an INN session before, and the sort of response that we had and to the arguments that we made that the antibody that we were representing was humanized basically came down to the fact that it was only a name, and the name has no effect on the function and the ability of the antibody to become a therapeutic. And you can level that argument back and say, if it's only a name, then why do we need it in the first place? So I think that there was very much a view that what the INN said was law, and we kind of just had to accept that.
John.
So I think we're all agreed that the naming subsystems are a pretty blunt instrument to try and describe something quite complicated, and so even if the move is towards ditching it altogether, I think the ultimate goal was a good one of trying to understand immunogenicity. So I just wonder if somebody could comment on the likelihood of there being some system in the future maybe that numerically assigns or makes an attempt to define immunogenicity, which is a thing that everybody's interested in. Maybe Matt could comment on that.
Well, I would say that that would be extremely difficult to do. Obviously, as Paul alluded to, the kind of immunogenicity side, sequence is only one aspect of that, and there is so much more, including pharmacology, formulation, manufacturing, and a lot of other stuff that we don't even know about that will contribute to immunogenicity. So just based on sequence definition, it's going to be very difficult, I would say impossible, to attribute immunogenicity in sequence like that.
If I could add one comment there, I mean, I think this might be the expectation of people not so close to this, that this is what the name suggests. And I think if one just follows this whole system uncritically, one would actually sort of support that this name is what is actually conveying this message. I don't think it does, for the reasons we have just discussed there, because the sequence, and certainly not the percentage, is any clear answer of the likelihood of immunogenicity, because of all the different complications in this. And I think one should, as a community, probably say that realizing that this is not what the sequence will say, we should certainly not create the impression that it does. And this is the reason for certainly my vote to get rid of the substem.
Thank you. John.
Yeah. So I'll be the first to agree. I think dropping the substems makes a lot of sense. My only concern is it's easy to sit here as a group and assert that we all agree on that. I don't know what kind of negotiation position we're actually in and whether or not we actually have any leverage. Right? So I think some thought should be put into fallback positions, such as maybe the synthetic substems, or even at least making new percent cutoffs so at least the legacy names will actually all pass the criteria, so there's some consistency, right? I mean, I think we're being a little overconfident that we can just dictate the way things work.
Well, I don't think we are being overconfident, because I think that there's this is kind of a confusing situation. However, I do think there is kind of an opportunity that to kind of mobilize ourselves as a community and to kind of engage the WHO, not who, in a productive and collegial way, and I think we will have much more impact if we do that with our sort of many organizations are participating, rather than if our individual companies sort of approach who about their specific instance. So I don't think we should assume success. But I think that we should kind of go in a constructive way, and maybe next April really is the first opportunity to do that. Currently, it's only scheduled for 90 minutes, and that seems like enough time to explain that there is a significant issue, and that there's kind of hopefully kind of broad consensus amongst antibody drug developers that this changes aren't working, and really to begin a dialogue as to what a robust system looks like that actually really does meet the stated goals of the INNs, that they're unique and that they facilitate our communication and don't kind of create a lot of confusion.
Yeah. And one other comment, and I think some consideration will have to be made for the unfortunate names that got caught in 2015, 2016 with a poor designation.
Thanks, John. Next question or comment?
Yes. I kind of agree with the numbering year type name. If we think about it, even to leave the map suffix, we'll probably be discussing this again in I don't know, 5 years. Is an antibody without effector functions an antibody with the hints modified? I mean, it's very difficult to keep track of all the mutations we can introduce in the FC that is not even taken into consideration for the names. And that's my comment. And the question that I have is, what's next? So it is kind of related with what we were discussing in the previous question, but what next? What we can do? The idea is to write a letter from this town hall and let them know that we are discussing this, or what we can do?
Yeah, I mean, it's a great question, and I think it's something that we've been sort of thinking about and sort of wrestling with. What is our most constructive way to engage the WHO in a way that's going to be sort of well received. And I think that the meeting in April, if there are sort of multiple kind of groups that approach around this issue, we can basically share the manuscript from MABS, and we can provide evidence that it's really that there's really sort of broad perception amongst the stakeholders that the system is broken, that I think you get to the point at least where you can sort of begin a dialogue as to what to do next. Another comment you made around just, essentially was around kind of future-proofing, and I think that's an important point. I mean, at this meeting, there are many sort of permutations where antibodies or antibody domains are fused to other proteins, maybe engineered scaffolds. Should those be considered as MABs? Maybe not. Have to decide really, what is the value and the purpose. You could make the analogy of our sort of taxonomy of sort of naming sort of new organisms. The challenge is that with our sort of the molecular world, that the future will sort of diverge in ways which are difficult to predict, and really just the sort of the imagination of ourselves and colleagues, sort of around the world. And I think that the proliferation of different formats will continue, and I think that makes some kind of developing a systematic naming system exceedingly difficult. So I think the kind of what Jim Larrick presented, which was just sort of a sort of a year of graduation with a number that you can sort of look up to see what it is, I think kind of resonates with me.
Just a quick comment, actually,
Please.
Paul, because if you look on the AMA rules, it does say rules for a monoclonal antibody. So technically speaking, using a substem system for bispecifics falls outside the definition of a monoclonal antibody.
Another inconsistency. Carrie.
Well, I agree that anything we say is going to be wrong because you can't capture everything. And I think it's quite hard to get all the data that's there, because the only thing that's really going to tell us about immunogenicity, if that's the issue, is what happens in the clinic. And it's actually quite hard to get that data. It's possible. I mean, I've spoken to people, John Lambert, if he's here, that resurfaced antibodies might even be less immunogenic than humanized antibodies. But where's that information? It's hard to get. So I think rather than make any value judgment on a number, we could say that antibody has to have its percentage, a number like X, with the nearest germline, the nearest germline, whatever that germline might be. I think Jim's idea of the year is great. And then a commitment to have other information there so that when companies put things in the clinic, they have to fill in information about how immunogenic was that, and what sort of antibody was it, and what the sequence is. Because unless we've got that data, in the future we can't think how is a new person coming along going to make a new antibody with all the information that's out there, because it's almost impossible to get. So if there was that commitment and a commitment to make that database, and we just started out with a simple number, and the nearest to the nearest germline and the year it was made, and then, as Jim says, you just click on something and it goes to all the information. The companies have to put that information on a database. Then we could start moving forward, and then looking back, we'd be able to see, well, okay, this percentage difference didn't really matter, but this particular thing did, because there was this aspect, I don't know, but we haven't got that information, so how can we classify? Everything we do is going to be wrong. Yeah, sorry to go on.
Thank you, Carrie.
Just a brief comment. That already exists, but I think you said something very valuable, which is the zoo, Z, all that stuff has an implicit right or wrong value system.
Yeah.
And that, I think, that is really the fundamental problem.
Yeah.
I mean, all the drugs have names. In fact, fluoxetine, that's Prozac. Rosuvastatin, that's Crestor. Unpronounceable, especially for me. But I mean, that already exists, and all those names, they have some description of what they do. As I don't know what pembrolizumab means, but you have the Li and the Tu and all the tumor, all that stuff that's been described, and I think that's valuable. I think by removing the stem, we get rid of this value system, and basically we treat monoclonals just like any other drug.
Yeah, we have to do that.
I think Jim, of course, is a radical person, but I kind of like the idea, but it's very radical and it's very different from every other medicine. I don't think that's going to work.
So I really enjoyed this week here, and I really learned a lot. But may I ask in this audience, who here is actually infusing an antibody into patients? Is there anybody here who is giving antibodies to patients? Personally? As a physician, prescribing them?
We have one here, Jim.
Who? One, well, at least. So let me tell you, I'm doing this. It doesn't make a difference what kind of drug you're infusing. Is it glycosylated? Is it chimeric? Is it fully human? I think it's not, it may not be the right thing to do to, I cannot tell you what is the right nomenclature, but I think it's definitely not the right thing just to say that anything that binds to something is a map. I don't think that does the job. I mean, you're all driving cars. Somebody is driving a diesel, somebody is driving a hybrid, somebody is driving an SUV. There are many names for this. There are drugs. I mean, there are so many drugs on this market, and physicians have to learn them all. Whether it's a beta blocker or a calcium antagonist, that's the difference. They have different names, and that is good for a purpose. So I think it is probably the duty of this audience, and maybe of some others, to really think hard how to categorize these binding agents and how to best describe them. But I think it's not just trivial, or it's not correct just to say, okay, it's so complicated, we just call them all maps. I don't think that will do it.
That's kind of a very interesting comment. Maybe I can sort of ask you, a question, or maybe kind of pose it to really everyone here. Really, what is the purpose of these names? Beyond providing a unique identifier, because kind of the direction that we head as a community will really be shaped by the purpose that you want to apply the names to. And I think Kerry made some really insightful comments that, it's very difficult to sort of future sort of proof, and so maybe the name should be kind of simple, because it's just and then you use some other mechanism, such as both kind of Jim and also Kerry sort of commented on to capture their complexity. Do you have a specific thought in mind or a suggestion as to, rather than just say, okay, not just kind of call them all MABs, do you have something specific in mind that you would propose instead?
Well, there are patients that receive these antibodies every week, every month, or several times. So it does make a difference whether they are fully human, or they are very close to human, or whether they are glycosylated or expressed in insect cells. I think this is something really, in a distinguished audience like here should work on. And I think, okay, whether 85 is the right cutoff or not, this is bullshit. But I think how an antibody was derived and how similar it is does make a difference, especially if you infuse it repeatedly into human beings. There is no doubt about this. And this has to be somehow represented in the name. How to do this exactly, that is a matter.
Thank you. Let's
Andreas has a comment right now.
If I may comment on that, I think, at least as far as I understand my colleagues, no one here would say that names should become totally random. The only thing, I think the best thing is to do the analogy between the INN name for small chemicals and for antibodies. So take an INN name for a kinase inhibitor, lapatinib, for example. The last two syllables say this is a kinase inhibitor, sure, and the first syllables are basically chosen by the company. Now, if we take an example like trastuzumab, the map is clear. It's the zoo that we're haggling about, and the two is the function, and the trust is the invention of the company. So I think the only thing that is at the debate here is the substem, the second to last syllable, because I think there is an agreement that the classification doesn't work on a grayscale. That's essentially the thing. The function, whether this is, for example, for tumor indication, for other things, these are syllables that should probably be maintained.
Mm-hmm.
And so if we decide that whatever classification we do is wrong because things don't fit, then perhaps one should simply say the second to last syllable, the substem, is up for debate.
I think it's up for debate how it should be defined, but I think it's not for debate to be dropped. That's my personal opinion. I think it is how to define this, I think, can be discussed.
So are you of the opinion that a system can be found that will fulfill all conceivable engineering tricks?
That is not the question. I think that there are systems to come close enough to differentiate, and I think that is up to debate. I think it's, in my personal opinion, as a physician who treats patients, I think it's not a debate just to drop this completely. I think it should be classified how the antibody was derived, how specifically. That is probably a matter to be discussed. There cannot be endless second initials, but there can be quite a few.
Thank you. The hour is late and the wine glasses are getting lower. So we will take 2 more questions.
Hi, I'd like to give the opinion of a relative outsider. This is my first time at this meeting, and I've only been working with monoclonal antibodies for a couple of years. But I have some things. I teach a lot of pharmacology. Names are important to students, to patients, to everybody. And I agree that the subsystem naming should be dropped because it's basically everything the same now. In fact, I would suggest you could even drop the M out of MAB because everything's monoclonal. But I would like to encourage some sort of a parallel system for the equivalent of a VIN number on a car or the EC naming for enzymes that allows you to specify in a long string that can be looked up by the year that it was approved, all the quantitative information, like what's the percentage of it of the identity to a human sequence, or is it, what format is it? So you can have subsections of that long string of numbers that totally defines that particular monoclonal antibody. Now, in terms of the generic names that people use, that's another matter, and I would like to see more targets put in as prefixes to the name so you know both what it's targeted to and that it's an antibody from the name itself.
Thank you for your comments. The last question or comment goes to John Lambert.
So my comment, the speaker before last, as a physician, raised some issues which caused me to get up. So just as an example, daratumumab was just approved, and it's a fully human antibody from Genmab. Now, Immunogen also developed an anti-CD38 antibody that was humanized by the resurfacing method that we developed in collaboration with Tony Rees. It's a Sanofi compound. And to my knowledge, it's as non-immunogenic in humans as daratumumab. However, it will be, I'm not sure if it's named yet, or if it has, I'm not aware of it, but it will be an ximab because it's resurfaced. And so Sanofi is already at a competitive disadvantage by being second, but they'll be potentially further disadvantaged by the perception that this is an immunogenic antibody. And it's not. So I would also support that. In fact, Andreas' comments about lapatinib, I mean, there are all these tyrosine kinase inhibitors. They're very different chemicals, but they all have the same root. I mean, so have a MAB as a root seems to make a lot of sense to me.
Thank you, John. Let me close by thanking you all for staying late and your active participation. To thank our panel for their input, and they're just